The Rotterdam Study is one of the largest and longest-running population health cohorts in Europe. Launched in the Netherlands in 1990, it has tracked thousands of adults in the Ommoord district of Rotterdam across decades, measuring diet, lifestyle, genetics, and health outcomes. Among its many publications, a 2004 paper in the Journal of Nutrition by Geleijnse, Vermeer, Witteman, and colleagues produced one of the most discussed findings in cardiovascular nutrition research. Among nearly 5,000 participants followed for a median of 7 to 10 years, higher dietary intake of vitamin K2, specifically in the form of menaquinone, was associated with a 57% lower relative risk of fatal coronary heart disease and a 52% lower risk of severe aortic calcification. Vitamin K1 intake showed no such association. The difference between these two forms of vitamin K is what makes this study biochemically interesting. In this video, what the biochemical distinction between vitamin K1 and K2 is, what matrix GLA protein is and why it matters for arterial calcification, what the Rotterdam study found in detail, the limitations of this observational data, and 3 takeaways framed appropriately for adults over 55. Quick note before we continue, this is educational content based on published research, not medical advice. Talk to your physician before changing your regimen, particularly if you take anticoagulants such as warfarin, which interact with vitamin K compounds. Now, the study. Vitamin K is a fat-soluble vitamin, but the term encompasses 2 structurally distinct molecules with meaningfully different biological roles. Vitamin K1, or phylloquinone, is found primarily in green leafy vegetables and is involved principally in hepatic blood clotting factor synthesis. Vitamin K2, or menaquinone, is a family of related compounds with different side chain lengths, designated -4 Malawian kwaches through -13 Malawian kwaches. -4 Malawian kwaches is found in animal products including meat and eggs and has a short tissue half-life. -7 Malawian kwaches is found primarily in fermented foods, especially the Japanese fermented soybean food natto, and has a substantially longer half-life in the body, on the order of 3 days versus hours for -4 Malawian kwaches. The longer half-life means -7 Malawian kwaches produces more sustained activation of vitamin K-dependent proteins outside the liver, in peripheral tissues including vascular smooth muscle and bone. Matrix GLA protein, abbreviated MGP, is the central mechanism linking vitamin K2 to arterial calcification. MGP is a protein produced by vascular smooth muscle cells and chondrocyte. It requires carboxylation, a vitamin K-dependent biochemical modification, to become biologically active. In its carboxylate form, MGP is one of the most potent known inhibitors of vascular calcification. It prevents calcium phosphate crystals from depositing in arterial walls. In its undercarboxylated form, which occurs when vitamin K2 is inadequate, MGP is inactive as a calcification inhibitor. Studies using immunohistochemistry in human arterial tissue have demonstrated that undercarboxylated MGP is present at significantly higher concentrations in calcified plaques compared to non-calcified arterial tissue. This mechanism was known from biochemistry before the Rotterdam cohort analysis. The Rotterdam study tested whether the dietary exposure to menaquinone correlated with the clinical outcome. The Rotterdam study design. This was a prospective cohort study. Participants were community-dwelling adults aged 55 and older recruited in the Netherlands in the early 1990s. Dietary vitamin K intake was assessed at baseline using a validated food frequency questionnaire, which is an established but imperfect dietary assessment method. Participants were divided into tertiles based on vitamin K2 intake. The primary outcomes were fatal coronary heart disease and aortic calcification severity, the latter assessed using chest X-ray scoring of calcium deposits visible on standard imaging. Follow-up extended to a median of 7 to 10 years depending on the specific analysis. The analysis adjusted for known cardiovascular risk factors including age, sex, smoking, body mass index, physical activity, and energy intake. The results. Participants in the highest tertile of vitamin K2 intake had a relative risk of fatal coronary heart disease of 0.43 compared to the lowest tertile, representing a 57% lower risk. This association was statistically significant. For severe aortic calcification, the highest K2 tertile had a relative risk of 0.48, a 52% lower risk of severe calcification, also statistically significant. Crucially, vitamin K1 intake showed no association with either outcome after adjustment for confounders. This K2 specific effect is biochemically consistent with the differential tissue distribution of K1 versus K2 and the relative importance of peripheral vitamin K-dependent proteins like MGP in vascular tissue versus the hepatic blood clotting role of K1. The mean vitamin K2 intake in the highest Rotterdam Study tertile was approximately 29 micrograms per day of menaquinone. To put that in dietary context, a typical Western diet provides roughly 5 to 15 micrograms of menaquinone per day. Foods high in -7 Malawian kwacha include natto, which contains the highest known dietary concentration of -7 Malawian kwacha, along with fermented hard cheeses such as Gouda and Edam, which were more commonly consumed in the Dutch population in which the Rotterdam study was conducted, and to a lesser extent egg yolks and fatty fish. The population in the Rotterdam study had a specifically cheese-heavy dietary pattern contributing to their K2 intake, which is worth noting when extrapolating to other dietary contexts. Now for the critical analysis. The Rotterdam study is observational evidence. This is essential context before interpreting the 57% risk reduction figure. Observational cohort studies cannot prove causation. People who consume more fermented foods and cheese may differ from those who do not in multiple ways that influence cardiovascular risk, beyond any direct effect of vitamin K2. The food frequency questionnaire method has known limitations, including recall bias and measurement error. The estimated effect sizes in this study are large by epidemiological standards. Large effect sizes in observational research sometimes reflect residual confounding, the healthy user effect, where people with healthier overall diets consume more K2-rich foods and also have lower cardiovascular risk for other reasons simultaneously. Second limitation, the aortic calcification measure, chest X-ray scoring, is a cruder measure than coronary artery calcium scoring by CT scan. It detects only relatively advanced calcification visible on plain X-ray. The mechanism via MGP suggests K2 effects would operate earlier, before calcification becomes radiographically visible, meaning this measure likely underestimates the biological effect at the calcification stage being modulated. Third, while subsequent mechanistic and interventional research has confirmed that vitamin K2 supplementation reduces undercarboxylated MGP levels in blood, and some smaller trials have shown effects on arterial stiffness, large-scale randomized trials specifically testing vitamin K2 supplementation on cardiovascular events in humans are limited. The observational association is biologically plausible and supported by mechanistic data, but the interventional evidence is less robust. A specific and critical safety note: vitamin K compounds, including K2, can interact with vitamin K antagonist anticoagulants such as warfarin and acenocumarol. These medications work by blocking vitamin K-dependent clotting factor synthesis. Any change in vitamin K intake, dietary or supplemental, can alter anticoagulation effect and change INR levels in patients on these medications. This is not a theoretical concern. It is well-established pharmacology. If you are on any anticoagulant medication, discuss any dietary change involving K2-rich foods with your prescribing physician before making changes. This is one of the more clinically important interactions in this area. 3 takeaways. First, the dietary sources of vitamin K2, particularly -7 Malawian kwacha, are specific and not evenly distributed across food groups. Fermented foods, especially aged hard cheeses, are the most practical dietary sources for a Western adult. Natto is the highest concentration source but is culturally unfamiliar to most North American and Northern European audiences. If you are interested in examining your dietary K2 sources, reviewing your intake of fermented dairy with a registered dietitian or your physician can be a reasonable starting point, with the anticoagulant interaction caveat firmly in place. Second, undercarboxylated MGP is measurable in clinical labs, though it is not routinely ordered. It serves as a functional marker of vitamin K status specifically in vascular tissue. Some research contexts use it as an intermediate biomarker. Whether it is clinically actionable in routine practice is not yet established. If you are seeing a physician who specializes in preventive cardiovascular medicine, asking whether undercarboxylated MGP is part of their biomarker assessment is a reasonable question grounded in the mechanistic literature. Third, the Rotterdam study results are hypothesis-generating rather than practice-changing in isolation. The mechanistic evidence for MGP's role in arterial calcification is strong. The observational association in Rotterdam is consistent with that mechanism. But for dietary or supplemental vitamin K2 to be incorporated into your cardiovascular risk management strategy, it needs to happen through a conversation with your physician who can weigh your full cardiovascular risk profile, your medication list, your dietary baseline, and the current state of the interventional evidence. The scientific question is live and being actively studied. It is not settled in either direction. To summarize, the Rotterdam study found that dietary vitamin K2 intake, not K1, was associated with a 57% lower risk of fatal coronary heart disease and 52% lower risk of severe aortic calcification across a median follow-up of 7 to 10 years in adults over 55. The mechanism involves matrix GLA protein, a vitamin K2-dependent vascular calcification inhibitor. This is observational evidence with biological plausibility but requires randomized trial confirmation. Vitamin K compounds interact with anticoagulant medications. This is educational content, not medical advice. Always consult your physician before changing your diet or regimen. Studies are preliminary, individual responses vary. Link to our free biomarker guide is in the description. New evidence reviews every Tuesday and Friday.